Good day everyone! Today we will be discussing the experiment 10 which is about collecting DNA. I am Phoebe Leal and here are my group mates to join me, Reina Andalahao, Vanna Costa, and Guevarra. For the content of our report, we will be starting with the introduction, followed by the objectives, methodology, discussion of results, conclusion, and recommendation. The references used in this report will be posted at the end of the video. So, what is DNA and how do we extract DNA from fruits? These questions will be answered in the course of the video. Keep an eye out because we are about to begin.
For introduction, let's first question, what is DNA? Deoxyribonucleic acid or DNA is a molecule that contains biological instructions that make each species unique. DNA, along with the instruction it contains, is passed from adult organisms to their offspring during their reproduction. DNA is a two-stranded molecule that appears twisted, giving it a unique shape referred to trusted source as the double helix. It is a long molecule that contains our unique genetic code. Like a recipe in book, it holds the instructions for making all the proteins in our bodies. Chromosomes are tightly coiled strands of DNA. Genes are sections of DNA that code individual proteins. DNA also carries important genetic information necessary for the survival and function of all life forms on Earth, and it is considered the master plan for all life on Earth. DNA is made of chemical building blocks called the nucleotides. The four types of nitrogen bases found in nucleotides are adenine, thymine, guanine, and cytosine. These building blocks are made of three parts: a phosphate group, a sugar group, and one of four types of nitrogen bases. To form a strand of DNA, nucleotides are linked into the chains, with the phosphate and sugar groups alternating. Adenine pairs to thymine, guanine pairs with cytosine. The order or sequence of these bases determines what biological instructions are contained in a strand of DNA. Next, according to Suleiman 2023, DNA carries the instruction for the development, growth, reproduction, and functioning of all life. All living organisms have DNA. It is basically the blueprint for everything that happens inside the organism cells. Overall, DNA tells an organism how to develop and function, and is so important that this complex compound is found in virtually every one of its cells. All living organisms are made up of cells that have DNA as the hereditary material, even in fruit. Every living organism has DNA. This includes plants and parts of plants like fruits, but also animals, bacteria, and fungi. All food from plants or animals contains genes. In cooked or processed food, most of the DNA has been destroyed or degraded and the genes are fragmented. Whether fresh or cooked, when we eat food, we digest it into its constituent parts from which we make our own genes and proteins. The ability of extract DNA is of primary importance to studying the genetic causes of the disease and for the development of diagnostic and drugs. It is also essential for carrying forensic sciences, sequencing genomes, detecting bacteria and viruses in the environment, and for determining the paternity. Trivia: who first discovered the DNA? The discovery of DNA in 1869 is credited to Swiss scientist Friedrich Miescher. Many people believe that American biologist James Watson and English physicist Francis Crick first discovered the DNA in the 1950s. In reality, they are not the first. Rather, DNA was first identified in the late 1860s by Swiss scientist Friedrich Miescher. Then, in the decades following Miescher's discovery, other scientists' research efforts that revealed additional details about the DNA molecule. Without the scientific foundation provided by these pioneers, Watson and Crick may never had reached their groundbreaking conclusion of 1953. Overall, thanks to them because they discovered the genetic and blueprint of our cells. Next, for the objective of our experiment first is to use some physical and chemical processes to extract a visible mass of DNA from the fruit tissue. Second is to discover which among the fruit samples produces more DNA or deoxyribonucleic acid. Third is to allow students to isolate and observe the DNA found in the fruits. And fourth, to contrast DNA from the chicks and from the fruit.
Before we proceed with the methodology, let us first discuss the rationale of this experiment. DNA is a double helix that provides the information required for an organism to develop, survive, and reproduce. According to biotechnology, the capacity to extract DNA is critical for researching the genetic causes of disease and developing diagnostics and treatments. It is also required for forensic science, genome sequencing, detecting viruses in the environment, and determining paternity. The DNA extraction principle was used in ripe fruits to determine and examine how DNA appears in fields in diverse fruits. This knowledge can be used to improve crops' resistance to disease, insect invasion, and climate change. The experiment was also conducted to demonstrate how DNA may be isolated from cells using fruits as illustrations. Furthermore, as the experiment progresses, the utilization of the substances used in DNA extraction, such as the detergent, alcohol, and salt, will be explained. Now we have the methodology. Under these are the materials used and the procedures done to conduct the experiment. For the materials, we have the fruit samples. In the experiment, we extracted the DNA from avocado, banana, grapes, mango, orange, and corn. We also used dishwashing soap, cloth, tap water, measuring spoon, 3 glass containers, isopropyl alcohol, strainer, salt, and Ziploc bag. Following the materials is the procedure. The procedure started with the preparation of the extraction mixture. The extraction mixture was prepared by pouring 50 ml of tap water into the beaker. Following that, 15 ml of dishwashing soap and 5 ml of salt were added. The salt was added to break apart the cells from each other and to neutralize the DNA charge and make the molecule less hydrophilic, meaning it becomes less soluble in water. The salt also helps to remove proteins that are bound to the DNA. The dishwashing soap, on the other hand, was used as a detergent to help break down the cell walls and membranes that surround the DNA. It helps to dissolve the fats and other lipids that make up these structures, making it easier to access the DNA inside the cells. After adding salt and soap, the mixture was mixed until completely dissolved before being placed aside. Inside the Ziploc bag, the ripe fruit sample was crushed with hands and fingers. 5 tablespoons of the fruit sample were completely incorporated into the mixture. The extraction mixture was then filtered using a cloth and a strainer. A spoon was used to press the masked fruit against the cloth and strainer to properly filter the solution. After filtration, the solution was added with 50 ml of chilled isopropyl alcohol. We added cold ethanol to slow down the enzymes that could break apart the DNA. It is made possible due to its low temperature. After adding the ethanol, since it is less dense than water, it formed the layer on top of the fruit soup. The salted DNA is insoluble to ethanol, so it will begin to precipitate out of the fruit soup at the boundary between the fruit and the ethanol. In less than a minute, you will already see cloudy strings of DNA like this.
We are now at the most exciting part of our report, the discussion of the results. Here, we are going to see in tackle age group results, and later, we will be discussing how these groups were able to come up with their corresponding results. Without further ado, let us now go to supergroup number 1. Here, we can see the supergroup 1 results for experiment number 10. Attached are the pictures gathered by group 1 for their fruit sample, which is grapes. And if we're going to look at it closely, the solution turned into the color peach upon extracting the grape juice. But is group 1 able to extract the grape's DNA for this experiment? The answer is no. As you can see in these pictures, there are no floating-like filaments that are present either at the top or at the bottom of the cup. A floating filament is a clump of thousands of DNA strands that is visible during DNA extraction, that is sometimes seen when exposed to light, sitting either at the top or at the bottom of the cup. Even though the group is able to follow the procedure accordingly, their experimentation is unsuccessful. Now, we are going to look at supergroup 2's results. The chosen fruit sample of group 2 is orange. Now, do we see a floating filament? If we're going to look at the picture here on the right, you can see the floating filament that is captured and extracted by the group. It is not much, but after following the procedure, the group successfully extracted the orange DNA. Our third sample is corn, which was used by the supergroup number 2. Here we can see that something is floating at the top of the sample. Is group number 3 able to extract the DNA? No. Although there is something floating in the group's sample, that is not a DNA. It is most likely pollutants that occurred during the procedure or pollutants came from the products and the materials used by the group, including the dirt carried by the wind. And with that, the answer is supergroup 3 is not successful in extracting the corn's DNA. Our next stop is the banana sample, which is used by the supergroup number 4. Look at these amazing pictures gathered by the group. Here we can see that the group succeeds in extracting the banana's DNA by looking at that big clump of filaments. It is not even thousands of strands, it may be millions of strands coming from the banana sample. Supergroup 4 did so well in this experiment that we are able to extract huge amounts of DNA. Now we have our fifth sample, which is from a mango tree. Supergroup number 5 used mango as their sweet sample for the extraction of DNA. But if we're going to look at these pictures provided by the group, not only we can see the filaments, but there is also too much foam present in the top, making it hard to see if there is extracted DNA or not. If we're going to scrape the foam from the top, we also risk the possibility to scope out the extracted DNA sitting at the top. Even though the group is able to do the procedure accordingly, the group is not successful in extracting the mango DNA. Down to our last group and last sample, we have here an apple sample used by the supergroup number 6. Here, as shown on the pictures gathered by the group, we can see little amount of DNA strands or filaments sitting on the top of the pod. And with that, we can conclude that the supergroup number 6 successfully extracted the apple's DNA. Out of 6 groups, only 3 groups are able to extract the DNA from the fruit sample that was brought. Although all of the groups were able to follow the procedure accordingly, it is not the expected outcome for the extraction of DNA through food products. However, a negative result is still a result. But if we only have one procedure, how come only 3 groups are able to extract DNA, and why they vary in amount? The occurrence of human errors during the experimentation is expected as to why some groups are not able to successfully extract the DNA. Remember the mango sample? If we're going to look back, the sample is too foamy, and with this we can conclude that the group stirred the sample too much, and since we use dish soap, the formation of bubbles may occur if stirred too fast or too much. According to the University of Nebraska AG Biosafety, one thing that can affect the DNA extraction is the percentage of isopropyl alcohol used for the fruit sample. We can also look at the fruit sample itself. All fruits are subject to extraction. However, they just vary on the amount that can be extracted. It is said that both kiwi and onion are best for fruit DNA extraction, but if we're going to use an onion, it is better to do it in an open space since onion has a strong smell. Another thing that we can consider is the ripeness of the fruit. The riper the fruit, the more DNA can be extracted from it. That is why supergroup 4 is able to extract huge lumps of DNA unlike the others with minimal filaments. We can conclude that the banana sample is much riper than the other samples, which leads to a higher yield of DNA. That wraps up our discussion of reports. All congrats to all supergroups that performed the extraction of DNA from fruit samples.
DNA molecules are normally invisible to the human eye, and one way to see them is by extraction. In line with the extraction, a certain tissue will undergo physical and chemical changes that will allow it to become visible. DNA collection might be simple or challenging. By conducting the DNA extraction from a different sample of fruits and then comparing it to one another, different results were obtained. The sample fruits such as grapes, Mangoes, oranges, pears, apples, and bananas were mashed to extract DNA from them. Only half of the fruit from these sample yields DNA. These were the banana, orange, and apple fruit. Since these sample fruits are triploids, which means that each cell's nucleus contains 3 copies of each kind of chromosome, in comparison to other fruits, they might generate more DNA. It is observed that numerous possible errors occurred with the conduction of this experiment. Unbeknownst to the group of teams, half of the sample fruits resulted in undesired results, that there was no DNA extracted from these samples. Each step, procedure, and variable has a definite purpose. Alcohol precipitates DNA, causing it to concentrate. The salt promotes precipitation by reducing the charge on DNA, while the cell membranes are destroyed by liquid soap or detergent, releasing the DNA. The reaction takes place in the water, which is also utilized to reconstitute DNA samples. Acquiring inadequate DNA and sample contamination may have been caused by a variety of things, including human error, the quantity of DNA present in the specific fruit, the number of cells in the volume of fruit consumed, and the maturity of the fruit. Fruits need more effort to extract DNA from, since the fruit must be separated from other biological components and mashed up to release the DNA. When the DNA from the fruit samples and the DNA from the chicks were compared, it is observed that extracted DNA from the chicks is less than the DNA extracted from the fruit sample, because fruits has more copies of DNA chromosomes than human cells. It appears that DNA from the chick cells is clear, hazy, and thread-like, and seems to be a thin, cotton-like thread of mucus. On the other hand, the DNA from the fruit sample appears to be yellowish white and coiled up, which seem to be a fiber thread. Conducting this experiment, it is discussed that there was no extracted DNA from most of the fruit samples, and only the orange, banana, and apple fruit was where the team was able to extract some DNA. For this reason, some recommendations will help in avoiding the recurrence of undesired results. Number 1, for doing DNA extraction on fruits, it is important to be specific and careful in doing the procedure, no more and no less. The team should be able to perform exactly what is written on the procedure to avoid the occurrence of undesired results. Number 2, it is also essential to pick a fruit that is suitable for extracting DNA. Some fruits are low in DNA and some fruits yield high DNA. Example of these are strawberries and bananas. Number 3, make sure that the fruit is ripe enough before the extraction of DNA, as it lessens the potential for acquiring inadequate DNA for the experiment.
So here are the list of references used in this reporting. Thank you so much for listening and have a great day.